Hi, welcome to lesson 4.5. Today we'll be talking about arbuscular mycorrhizal fungi in soil and ecosystem disturbance. We'll look at what happens to arbuscular mycorrhizal fungi with mechanical and chemical disturbances, at things like tillage, addition of chemical fertilizers to soils, and then we'll look at how they respond to heavy metal toxicity, but also their involvement in being able to help plants remediate heavy metal toxicity from soil. We'll also look at arbuscular mycorrhizal fungi response to fire, as well as their interactions with invasive plants. So let's jump into lesson 4.5. Now that we've covered the whole plethora of information about the beauty of this symbiosis between arbuscular mycorrhizal fungi and their host plants, it is pretty obvious that if the plants are missing or disturbed or removed, that these arbuscular mycorrhizal fungi will no longer be present as well. So even though spores survive freezing, wetting, and drying, they are quite sensitive and not a very long-lived structure. So they always require a host plant following some time of waiting for that host plant to be present. They do actually require one to start its life cycle. So unlike, for example, bacterial spores, arbuscular mycorrhizal fungal spores are not as great as survival as bacterial spores, and even the ectomycorrhizal fungal spores, which we'll talk about in future lessons. Propagule survival is pretty much non-existent without a host. In host absence, very rapidly there is a decrease in the potential of hyphae or mycelium or root containing fungal propagules decreases with time quite rapidly. So basically that means that hyphae themselves that infiltrate the soil ecosystem beyond roots are highly susceptible to disturbance. So things like fires, topsoil removal, flooding, mining, forestry, agriculture, tillage, leaving the soil fallow for long periods of time, compaction, and production of non-mycorrhizal crops will all lead to diminished numbers or complete destruction of these fungi in soils. In very extreme soil situations, in cases where soil is saline, acid, the aridity is extreme, where there's waterlogging, and in case of severe climatic conditions, arbuscular mycorrhizal fungi might be absent. They're very sensitive to these kind of disturbances, these sort of soil aberrations. The physical disturbance of the top 30 centimeters of topsoil drastically affects inoculum potential short term. So this is specifically important in agriculture where the soil is turned through tillage and those hyphal networks are broken down basically, and as the spores are moved around further away from where they would normally sit in relation to a host plant, all of those disturbances taking out the plants that have been colonized by arbuscular mycorrhizal fungi, that basically destroys their ecosystem. Also, things like synthetic fertilizers like phosphate and nitrogen added also disrupts or even completely diminishes, gets rid of really arbuscular mycorrhizal fungi. When the plant no longer requires this symbiosis, the plant will not want to give sugar away when it's getting fed this direct synthetic compound. And the plant is literally tricked to remove its microbiome, remove its fungal partner, because it's receiving this fast food, this chemical that it recognizes as food that is basically force-fed in large amounts to the plant, and it no longer then requires this symbiotic partner that it has relied on for millions of years to feed it. So this really reduces arbuscular mycorrhizal fungal diversity and abundance, and residual phosphate inhibits colonization, even after conversion of agricultural practices from conventional to organic systems. That's how much synthetic fertilizers actually prevent these natural symbiotic plant partners to flourish and to exist, really. There is, however, increased plant survival in some contaminated soils, but not in others. So what this means is that some soils that have some contaminants in them might actually have adapted Arbuscular mycorrhizal fungi in them. And this is where it becomes really interesting in remediating poisoned or heavy metal enriched soils. Some of these Arbuscular mycorrhizal fungi are actually quite resilient, and in partnership with certain plant species, they will actually reseed or restart the ecosystem. They're kickstarted into succession again and allow remediation of these contaminated soils. Another fantastic potentiality of mycorrhizal fungi is that they can be used in this process of phyto or mycoremediation. And this is where we have contaminated soils. So you can have a look at the different types of phytoremediation. That's using a plant where the plant has specific ability to transform particular nasty chemical that's in the soil. Some Arbuscular mycorrhizal fungi can reduce heavy metal toxicity or heavy metal stress in plants. They might do this by reducing uptake and therefore protecting the plant, versus increasing uptake because these plants that they're growing with are actually quite tolerant to the heavy metal. But this means that the soil is detoxified when the plant takes it up. These plants are called the hyperaccumulators. Also, glomalin chelation has been reported, so that means glomalin actually binds some heavy metals, and even chitin, so the chitinous wall of the Arbuscular mycorrhizal fungi can bind certain heavy metals as well. In this case, the chelation means the heavy metals might not be taken up into Arbuscular mycorrhizal fungi, but if they do get taken up into the cell contents, into the cytosol, they can actually be pumped out by heavy metal transporters. That's to reduce the heavy metal concentration in the mycorrhizal fungi. In some instances, in heavy metal contaminated soils, there is greater colonization of Arbuscular mycorrhizal fungi and greater hyphal proliferation. So this increase basically protects plants from heavy metal toxicity. Spores from polluted soils versus non-polluted soils actually show reduced cadmium, copper, zinc, and manganese uptake. So the spores themselves are adapted in polluted soils to not take up the heavy metals. So heavy metal hyperaccumulators, which are plants that actually love taking heavy metal in and being okay with that, in conjunction with tolerant Arbuscular mycorrhizal fungi, can be used to increase heavy metal content in tissue of plants and therefore remediate soil from the heavy metal burden. And you can remove those plants, remove the heavy metal from the land by just growing these hyperaccumulators with their tolerant Arbuscular mycorrhizal fungi. So this is a huge area of research, and it's fascinating, and it's just a lovely way to clean up the environment using plants and their associated Arbuscular mycorrhizal fungi. It is very important to mention that Arbuscular mycorrhizal fungi rely heavily on nutrients from the leaf litter that falls down to the ground. They basically receive those nutrients from those bacteria and other fungi that are actually living and decomposing the leaf matter. When that debris is being decomposed, they pick up those nutrients and pass them on to the plant. So when we do litter burn off, we actually reduce nutrients for mycorrhizal fungal development and growth. And by doing so, we also reduce, obviously, abundance of other fungi such as the saprotrophic or the decomposer fungi and plants. And of course, heat directly kills hyphae and spores. So burning is not a great idea when it comes to fungi, especially the Arbuscular mycorrhizal fungi. Even though they might survive deeper in the ground and the heat might not penetrate that far, a lot of the inoculum, a lot of the hyphae, they actually feed at the leaf litter level. So burning is always going to reduce Arbuscular mycorrhizal fungal load in the ecosystem. They are also facing climate change. And it's an interesting concept to think about the fact that plants photosynthesize and their food in photosynthesis is carbon dioxide. So one would think that when there are elevated carbon dioxide levels in the atmosphere, that the plants would just photosynthesize more and therefore feed their fungi more on their roots, and therefore the ecosystem could draw down more carbon into the soil and plants would grow better because the systems are kicking in with this greater abundance of food, really, from the atmosphere, the carbon dioxide. And this is probably true, and there is some evidence that this could be possible. So the idea is that more carbon dioxide means greater plant growth, greater arbuscular mycorrhizal fungal biomass, via greater photosynthesis, and therefore more stimulated colonization and hyphal production, and therefore more exudates running to the plant roots, and therefore more necromass or dead fungal mass being deposited, and more chitin from their cell walls being deposited, and as a result, greater aggregation because of all these factors mentioned, and therefore more carbon storage. So that is what is possible. This is all, however, temperature and moisture dependent. When there is no moisture because there's not enough vegetation in the ecosystem and the temperature rises continually, of course, this is going to very much affect this process. So the best thing to do really is to just grow more plants, regardless of what kind of plants they are, to return at least the water cycle back, especially in our arid country like Australia. We need to be able to get that moisture flow again, where past the mountain ranges moisture doesn't come in anymore and farms are left in drought. So we need to actually balance this with enough biodiverse vegetation and create that moisture again and create the layer of organic matter to hold that moisture in. So this is definitely a thing where we can reverse climate change using these systems like the arbuscular mycorrhizal symbiosis. However, we need to provide the conditions for that to actually stimulate that process. And this is, of course, also species and species host dependent. So different species of arbuscular mycorrhizal fungi and their hosts respond differently to this climatic change that's happening. There are a couple of great reviews about carbon sequestration through arbuscular mycorrhizal fungi, and you can just read them here and look them up. And also this paper here, that it's quite recent, it's the potential of arbuscular mycorrhizal fungi in carbon cycling, a review. So you can look that up and have a read as to this whole process where arbuscular mycorrhizal fungi, given the right conditions and giving the right food from their host plants, can actually increase aggregate stability and deposit that chitinous cell wall to the ground, to the soil, and that soil organic matter can be protected and recalcitrant products made by the fungi, and then carbon sequestration might be possible. And of course, there's also our famous glomalin molecule there as well. So the other thing we need to point out is that there are a lot of species that are being introduced around the world. Species travel, and of course we know our beautiful lantana, how much that proliferates in certain conditions in Australia. And of course there are dynamics between arbuscular mycorrhizal fungi and invasive plants. And these plants normally come into new or disturbed habitats, and they really do alter native arbuscular mycorrhizal density and diversity. They also affect native plant communities as a result. Because some of these plants that are invasive, they might have their own arbuscular mycorrhizal fungi on their roots, or they might partner up with the arbuscular mycorrhizal fungi of the native kind, the native species of arbuscular mycorrhizal fungi, and this would mean that they are supported, their growth is supported, they're doing really well, and they just keep invading the ecosystem because that symbiosis works really well for them. Other forms of disturbance through invasive plant species is an example here is the garlic mustard plant that comes from Europe, and it's a non-mycorrhizal, non-arbuscular mycorrhizal plant, and it's in the family Brassicaceae, and these guys actually don't like mycorrhizal fungi, so they're one of the families that doesn't prefer to be in this symbiosis. And what it does is it actually leads to biochemical inhibition of native North American arbuscular mycorrhizal fungi. So they basically don't have arbuscular mycorrhizal fungi on their roots, they don't like them, so therefore they pump out this biochemical to inhibit the arbuscular mycorrhizal fungi forming a relationship with them. And so, of course, this chemical then affects native plant communities. And it can be quite adverse because it's basically pretty much suppressing arbuscular mycorrhizal fungi formation and growth. However, arbuscular mycorrhizal fungi in its native soils are actually resistant to this garlic mustard inhibition of arbuscular mycorrhizal fungi. So basically, where plants have evolved together in a community where some don't like being mycorrhizal and some do, and to protect themselves from the mycorrhizal fungi, these plants that are in these native communities that have been then adapting to the ecosystem for long periods of time, they actually don't have an effect on the arbuscular mycorrhizal fungi. So the arbuscular mycorrhizal fungi are basically okay with that chemical that the non-mycorrhizal plant is producing. But of course, if you introduce these plants to sensitive environments where these arbuscular mycorrhizal fungi haven't been exposed to these allelopathic compounds, these compounds that inhibit arbuscular mycorrhizal fungal growth, then it's going to have a detrimental effect on the ecosystem. Pre-established arbuscular mycorrhizal fungal symbiosis, however, ameliorates this plant's allelopathic effect. So if we have a nice ecosystem and this invasive plant comes in and the ecosystem is resilient, there's lots of interactions happening and there's a lot of plants that are mycorrhizal and everything's working, there is not much chance of this garlic mustard or another invasive plant having huge detrimental effects. Everything sort of just gets dealt with because there's microbial diversity and these compounds are rapidly decomposed. And really, there's not much to it. But in disturbed ecosystems, under climate change conditions, all these things that have happened, particularly in this country, there's so much disturbance. There's so many introduced species. There's so many animals that are introduced that have hooves that Australian animals don't have. You can imagine the pressure, the compaction, everything's working against the natural arbuscular mycorrhizal ecosystem that we have in our soil. It's all kind of getting disturbed constantly and continually. So when invasive species are added on top of that, now that creates very poor situation for plants, for the plant communities and the ecosystem and soil biodiversity. So this concludes lesson 4.5 on arbuscular mycorrhizal fungi and disturbance in ecosystems and in soils, and the potential of that remediation as well. And in the next lesson, lesson 4.6, we'll look at arbuscular mycorrhizal fungi in soil and ecosystem restoration. So we'll dive into a little bit more of that potential that they have in helping us to restore our ecosystems and our soils. Okay, so I'll see you in lesson 4.6. Bye for now.